Today, we're going to tear down a really common assumption, which is that taking a medicine is, a simple, fixed, and predictable process.
It's anything but.
Right. I mean, if you've ever had a doctor adjust your dose, or if you've ever taken a drug that worked wonders for your friend but did absolutely nothing for you, you already know, just instinctively, that this stuff is intensely personal.
That's right. When a medication enters your system, it's not a passive thing. It kicks off this incredibly dynamic interaction that involves four critical elements.
Okay, what are they?
First, you have the chemical properties of the drug itself. Then there's the pathology of the disease you're actually fighting. Third is the unique physiology of the patient you. And finally, the overall context or the milieu that the therapy is happening in.
And we've spent a lot of time digging into some really foundational source materials on this, looking at clinical pharmacology, the science of pharmacotherapy, and the really stringent demands of drug development.
So our mission today is, I think, pretty clear. We want to give you a shortcut to understanding the complex strategies that go into determining your drug dosage. We're going to expose the huge array of factors that make your response totally unique.
Everything from your kidney function to your genes.
Exactly. And then we're going to pull back the curtain on the incredible rigor, the sheer time, and, honestly, the expense it takes to bring any new medicines safely to market.
Okay, so let's start with the most basic concept of all, the dose. We think of a dose as this single, fixed number, but our sources just immediately shatter that idea. They remind us that the dose always has to be qualified by the chosen therapeutic response.
Yes. And the classic example to show how complex this gets is aspirin. Just plain old aspirin.
Which is just acetylsalicylic acid, chemically speaking.
Right. But the concentration you need in your body for a specific goal is radically different depending on what you want it to do.
Okay, so let's break that down. If I have a headache and I just need simple pain relief, what am I looking at?
That's the analgesic dose. You're looking at about 0.3 to 0.6 grams. This is a relatively small amount.
But then if the goal totally shifts, say to long-term prevention of a heart attack or a stroke, you're using aspirin for a different reason.
A completely different reason. You're harnessing its ability to prevent blood clots. That's the antiplatelet dose. And that dose just plummets down to only 60 to 150 milligrams per day.
Wow, that's an order of magnitude smaller.
It's tiny in comparison, but it's a completely different clinical intervention. And then there's a third scenario.
Which is?
If a patient is battling severe chronic inflammation, something like rheumatoid arthritis, the anti-inflammatory action requires huge concentrations.
So the dose goes up again?
It skyrockets, up to 3 to 5 grams per day. So you have the same molecule, 3 distinct goals, and 3 utterly different amounts.
So those standard doses you see on the bottle, they're really just based on population averages, right? They're for an idealized average patient.
Exactly. But, as we're about to explore, the core, inescapable truth of all of this is that almost no one is truly average. That variability is what demands that the dose has to be individualized.
Okay, so let's unpack the strategies that prescribers actually use to do this. I mean, it can't just be a guessing game. It has to be systematic.
It is. It is very systematic. The whole idea is that the dose you need is a function of the drug's potency. So the concentration you need at the target site and how your body is going to handle it, it's pharmacokinetics. The sources lay out 4 main strategies for this.
All right, what's the first one?
The first and simplest is the standard dose. This is the one-size-fits-all approach. It works for drugs where individual variation is pretty minor, or, and this is the key part, when the drug has a very wide therapeutic window.
A wide therapeutic window. So that means the toxic dose is way, way higher than the effective dose.
Exactly. There's a massive margin for error. It lets the prescriber give a dose that's big enough to work for almost everyone without risking toxicity. You see this with things like oral contraceptives, penicillin, or a lot of basic diuretics.
It simplifies things a lot.
Immensely.
Okay, moving up in complexity then, we have the regulated dose. Now, this sounds like it's continuously monitored, but the monitoring isn't actually of the drug itself.
That's a really crucial distinction. The regulated dose applies to drugs that modify a body function that is, itself, easily and objectively measurable.
By what?
Think antihypertensives, where you can just measure blood pressure, or anticoagulants, where you measure clotting time, or hypoglycemics, where you're measuring blood glucose.
So instead of tracking the drug in the blood, the doctor is tracking the effect of the drug on the body.
Correct. The effect is the best monitor you could ask for. If the blood pressure is too high, you increase the dose. If it's too low, you bring it down. You generally don't need to measure the plasma drug concentration because the body is giving you all the feedback you need.
But what happens when that response isn't easily measurable? Like, if a drug is acting in the brain where you can't just take a simple reading.
That brings us to the third strategy, the target level dose. This one is essential for drugs that have a narrow therapeutic index.
Meaning the effective dose and the toxic dose are really close together.
Dangerously close. So for these drugs, hitting a specific, known effective plasma concentration is absolutely vital. We're talking about drugs like lithium, certain anti-epileptics, or digoxin for heart failure.
So how does a doctor hit that narrow target without overshooting?
It's a two-step process. You start with an empirical dose, which is basically a calculated best guess to get you into that target range. Then you actively monitor the plasma concentration to see if you actually hit the mark.
And this is where therapeutic drug monitoring, or TDM, comes in.
This is its bread and butter. If the level is too low, you adjust up. If it's too high, you adjust down. Now, if you don't have monitoring facilities, which is a really poor compromise, you're forced to rely on just crude observations of the patient over a long time.
And finally, you have what sounds like a tightrope walk, the titrated dose. This one seems to imply there's a built-in conflict between getting the maximum effect and avoiding side effects.
This is the toughest clinical scenario. It applies when the dose you need for the maximal therapeutic effect will inevitably cause intolerable adverse effects. It's just a given.
So you have to compromise.
Yeah, it's a compromise. Optimal dosing here is all about striking a balance. You're aiming for a submaximal therapeutic effect, one that the patient can actually tolerate.
And there are two ways to approach that titration.
Yes, and it depends on how urgent the situation is. In most non-critical diseases, you use upward titration. You start low, you go slow, and you gradually increase the dose until you hit that sweet spot or the side effects become too much.
But in critical situations,
In life-threatening scenarios, you might use downward titration. You start with a high dose to get an immediate effect, and then you back off quickly as the adverse effects start to appear. You see this constantly with intense therapies, like anti-cancer drugs, or for managing something like Parkinson's with levodopa. It's a constant balancing act.
okay, so if individualizing one drug is that hard, what about these fixed dose combinations, the FDCs, the pills with 2 or more drugs in them? They seem convenient, but it sounds like they fly in the face of this whole principle of individualization.
They absolutely offer convenience, and that's their big selling point. You get better patient compliance, fewer pills to remember, but that convenience is only rational if the patient truly needs every single component in that pill.
Let's talk about the real pharmacological advantages first, though. When do they make sense?
Well, beyond compliance, they can provide true synergistic effects, where 1 + 1 = 3. The classic example is the antibacterial combo of sulfamethoxazole and trimethoprim, or combining levodopa with carbidopa, where the carbidopa helps reduce levodopa's side effects.
You also see them used to get additive effects, right, where the two drugs have different mechanisms.
Exactly. Like combining amlodipine and atenolol for severe hypertension. They're hitting blood pressure from two different angles. Or, as a third advantage, you can use one drug just to counter a side effect of the other one. Like combining a thiazide diuretic, which makes you lose potassium, with a potassium-sparing diuretic. It's a very clever way to balance things out.
And for really serious infectious diseases, these FDCs are sometimes essential.
Oh, absolutely critical. For things like tuberculosis, HIV/AIDS, or falciparum malaria, the combination formulas are there specifically to make sure the patient cannot take just one drug.
Because that would promote resistance.
Instantly. It's a public health necessity.
Okay, now let's get to the risks. This seems to be where the loss of individualization really hurts. What's the biggest problem?
The core problem is the inability to adjust the dose of the components separately. The ratio is fixed. So if a patient has, say, kidney failure that affects drug A but not drug B, you can't reduce the dose of drug A without also cutting the dose of drug B, which they might need at full strength.
And if the patient doesn't need all the components, they're just getting unnecessary side effects and paying for a drug they don't need.
Precisely. And it gets worse. What if the components have different time courses of action? One is short-acting, one is long-acting. A standard dosing interval, like twice a day, is going to be pharmacologically wrong for one of them.
And if a side effect happens, you have no idea which drug caused it.
Exactly. And if one component is contraindicated, the whole product is out. And our sources are crystal clear about one non-negotiable rule here, specifically with corticosteroids.
What's that?
Corticosteroids should never be combined with any other drug for internal use. Period. The principle has to be ruthless. If the patient doesn't need an ingredient, the combination is irrational no matter how convenient.
Which brings us to the rationality check. It feels like thousands of these FDCs are on the market, but how many are actually considered rational?
The numbers are, frankly, shocking.
Mm.
The 2017 WHO Essential Medicines List, which is the gold standard, only includes 25 FDCs that it deems rational.
Mm.
India's national list had only 20.
25
Yeah.
out of thousands. That's a dramatic reveal.
It is. It underscores the huge danger of all these non-essential combinations. It got so bad that India recently had to issue a blanket ban on 344 irrational FDCs just to protect patients from unnecessary risk and cost.
So that discussion on dose really leads us right into the next level of complexity. All the inbuilt biological variations that explain why your response to a drug is completely unique.
We have to move from just dose setting to understanding all the variables that determine how high the drug concentration gets in your plasma, how your receptors respond, even differences in your baseline hormonal tone.
And this modification can be quantitative.
Right? But changing the amount of action, which you can often fix by adjusting the dose. Or it can be qualitative, like an allergy or an idiosyncratic reaction, which usually means you have to stop the drug immediately.
Let's start with the most obvious factor, body size. A standard dose is for, what, a 70-kilogram average adult?
That's the benchmark. For people who are exceptionally large or small, the dose is often scaled using a simple body weight ratio.
But the sources argue pretty strongly for using body surface area, or BSA, instead of just weight. Why is that considered more accurate?
BSA is a better metric because it correlates more closely with fundamental physiological constants, things like total body water, extracellular fluid, and overall metabolic activity. The Du Bois formula for it is complex, but it reflects the body's actual physiological space better than just mass.
So why isn't it the standard everywhere, that complexity?
That's the practical problem. The formula is a pain to use in a busy clinic. So dose recommendations based on BSA are really restricted to high-risk fields, mostly anti-cancer chemotherapy and a few other drugs where that level of precision is critical.
Okay, if size is that complex, it must get even harder when we add the dynamic of growth. Let's talk about age, starting with pediatrics. The mantra here seems to be that infants are not just small adults.
It is a critical point that was learned the hard way. Infants have these physiological immaturities that completely change their drug profile. Take the kidneys. The GFR, the glomerular filtration rate, which is how fast blood gets filtered, is very low.
And what does that mean for a drug?
It dramatically prolongs the half-life for drugs that are excreted unchanged. For something like gentamicin or penicillin, the half-life can be 3 to 5 times longer than in an adult. The GFR doesn't even reach adult rates until about 5 months of age.
And I assume the liver, the body's metabolic engine, is also underdeveloped.
Absolutely. The hepatic drug metabolizing system is inadequate. The most tragic classic example of this is the antibiotic chloramphenicol. An infant just can't metabolize and detoxify properly, so it builds up, leading to circulatory collapse and death. It's known as gray baby syndrome.
That's a terrifying example.
It is. And on top of that, the blood-brain barrier is more permeable in newborns, which lets certain drugs get into the central nervous system at higher concentrations.
But absorption can also be different, sometimes even faster.
Yes, especially non-orally. Transdermal absorption is faster because their skin is thinner. And rectal absorption is surprisingly fast and predictable in infants, which is why liquid diazepam is given rectally to stop acute febrile seizures. You need a fast, reliable route.
So what happens after that first year? I was surprised to read there's a reversal.
It's fascinating. After about 12 months, a child's drug metabolism often becomes faster than an adult's. For some drugs, like theophylline, children have a shorter half-life and may actually need a higher dose per kilogram than an adult to get the same effect.
And kids can have very specific adverse effects tied to their development.
Right. We see corticosteroids suppressing growth, and the classic one is tetracyclines depositing and developing teeth, causing permanent discoloration and deformation. The drug profile of a child is a constantly moving target.
Now let's contrast that with the challenges of the elderly. Here, it's not about development, it's about decline.
The elderly population is probably the biggest pharmacological challenge we face. Renal function declines steadily. GFR is often cut in half by age 75. That means dose reduction is mandatory for any drug cleared by the kidneys.
Hepatic function declines as well, I imagine.
It does. Both the microsomal enzyme activity and the actual blood flow to the liver decrease. So you have this 1-2 punch of reduced renal and metabolic clearance, which makes the elderly highly susceptible to cumulative toxicity.
And what other systemic changes are going on?
Well, absorption can be slower. Plasma albumin levels often drop, which means less protein binding and more free active drug floating around. And the volume of distribution changes, which affects how different types of drugs are stored in the body.
And the risk of interactions must just be through the roof because of polypharmacy taking multiple drugs at once.
It's exponential. Elderly patients are often managing several chronic conditions, so they're on multiple drugs. This directly leads to a much higher incidence of adverse reactions, things like postural instability, giddiness, mental confusion, things that dramatically increase their risk of falling.
Moving on from age and size, let's look at more intrinsic differences. Starting with sex, besides the obvious size difference, the most complex area has to be pregnancy.
Oh yes. The physiological changes during pregnancy are marked and progressive. GI motility slows down, which can delay drug absorption. Plasma volume expands, which increases the volume of distribution for many drugs.
And protein binding gets really complicated.
It does. Plasma albumin falls, which increases the unbound active fraction of acidic drugs like warfarin. But then alpha-1 acid glycoprotein increases, which decreases the unbound fraction of basic drugs. It's a highly volatile environment.
We also see differences based on species and race. We know rabbits are resistant to atropine, but what about within humans? What kind of racial differences are documented?
These are differences in population averages, and they're often tied to genetic variances. For example, sources consistently note that Black individuals often require higher concentrations of drugs like atropine or ephedrine for pupil dilation, and beta blockers are often found to be less effective as antihypertensives in people of Afro-Caribbean descent compared to Caucasians.
This all leads directly to what might be the future of therapy: genetics and pharmacogenetics.
The variability in the dose needed for the same effect can be huge, like 4- to 6-fold between people. And this is because the major determinants of drug response, the transporters, the enzymes, the receptors, are all controlled by our genes.
So pharmacogenetics is the study of that genetic basis, and pharmacogenomics is actually using that information to guide drug and dose selection.
Precisely. And its predictive power right now is strongest for what we call monogenic traits, where the variability comes from a single gene. The classic example is G6PD deficiency.
Explain what that is and why it's so clinically important.
G6PD is an enzyme. People who are deficient in it suffer acute hemolysis. That's the rupture of their red blood cells if they're given certain drugs. It's a life-threatening reaction. The list of drugs that can trigger this includes primaquine, sulfonamides, and even high-dose aspirin.
So there are simple tests for this.
Yes. Simple, inexpensive spot tests are already used in practice in some places, but most variability isn't due to these single-gene defects. It's due to more subtle differences, called SNPs.
Single nucleotide polymorphisms.
Right. A substitution of a single base pair in our DNA. They're very common. And while we've collected massive amounts of pharmacogenomic data, the practical, routine application of it is still pretty limited.
Why? If genetics is so key, why isn't it standard practice?
It's a logistical issue, mostly. To fully use it, we'd need to do multiple drug-specific genetic screenings for every patient. However, since you only need to genotype a person once in their entire life, the potential for this to become the mandatory standard for guiding that initial drug and dose choice is just immense.
So one day we're just going to hand our doctor a full genetic profile before they write any prescription. It really does feel inevitable.
I think so.
Which is a great place to transition to how existing illnesses can change this whole equation.
Absolutely. Diseases aren't passive. They actively modify how your body handles a medication. A standard dose for a healthy person could be wildly toxic for someone of the same size who has, say, cirrhosis of the liver.
Okay, let's start with liver disease. The liver is the body's main metabolic hub. How does its failure mess with drug action?
The effects are multi-layered. First, for drugs that have high first-pass metabolism, meaning the liver normally chews up a lot of it before it hits the bloodstream, liver failure dramatically increases their bioavailability.
Especially with something called portocaval shunting. What is that?
In severe liver disease, blood finds detours around the scarred liver, bypassing it entirely. This means the drug avoids that filtering process, and a much higher concentration of active drug floods the body. You have to make massive dose reductions.
And a failing liver produces less albumin.
Right, which is a key binding protein. So there's less protein to bind acidic drugs like warfarin. This means more of the drug is in its free, active form, which increases both its potency and its toxicity risk.
So doctors have to be really careful with drug selection.
Very careful. For example, they'll often substitute diazepam, which relies heavily on the liver, with something like oxazepam, which is handled differently. And critically, the brain of a person with cirrhosis is much more sensitive to depressants. A normal dose of morphine could trigger a coma.
Okay, what about the other major clearance organ, the kidney? When we're talking about renal failure, we're talking about a huge drop in drug clearance.
A marked reduction. Any drug that's excreted primarily unchanged like aminoglycoside antibiotics or digoxin is going to accumulate. Its clearance rate is directly tied to the fall in the patient's creatinine clearance, or CL-ever.
So how does a doctor calculate the dose reduction based on that CLR?
The initial loading dose is usually the same, but the maintenance dose has to be cut proportionally to the fall in CL count. To give you a sense of scale, if a patient's CL count is between 50 and 70, the dose rate needs to be cut to about 70%. If it's severely low, say 10 to 30, you can only give them about 30% of the standard maintenance dose.
That's a huge reduction. It really drives home why the standard dose is meaningless in renal failure.
It's all about constant monitoring.
Yeah.
And on top of that, renal failure makes the blood-brain barrier more permeable, so CNS depressants have a stronger effect. Pethidine, for instance, has to be avoided because its metabolite accumulates and can cause severe seizures.
And some standard drugs just become dangerous.
Thiazide diuretics become ineffective and actually worsen the uremia. And potassium-sparing diuretics are completely contraindicated because they can cause life-threatening hyperkalemia.
And we see these surprising connections in other diseases, too.
Definitely. Take congestive heart failure. Drug absorption decreases because of gut edema, and elimination slows down because of poor blood flow to the liver and kidneys.
Right.
Or thyroid disease. Hypothyroid patients are more sensitive to CNS depressants, while hyperthyroid patients can be resistant to some actions of digoxin, but more prone to others.
The disease itself dictates the pharmacology.
It really does.
Now let's talk about variations that happen during therapy. Accumulation seems like the easiest to understand. The drug goes in faster than it comes out.
Exactly. It's a slow burn toxicity. For slowly eliminated drugs, the risk is very high. Prolonged use of chloroquine can accumulate and cause retinal damage. You have to be very careful with scheduling.
Then there's tolerance, which is where you need a higher dose over time to get the same effect.
And tolerance can be acquired, or it can be natural. But what's really crucial is that tolerance does not develop equally to all actions of a drug.
What's the key clinical example of that?
Morphine. Tolerance develops very strongly to its pain-relieving and euphoric actions, but tolerance develops much less, if at all, to its constipating and pupil-constricting effects.
And what's happening in the body to cause that tolerance?
Two main things. You can have pharmacokinetic tolerance, where the body gets better at eliminating the drug.
Yeah.
For example, barbiturates induce liver enzymes that speed up their own metabolism. Or you can have pharmacodynamic tolerance, where the target cells themselves become less responsive, often by reducing the number of receptors on their surface.
And what about tachyphylaxis? This is a rapid form of tolerance.
Extremely rapid. It's tolerance that develops when doses are repeated in quick succession. You often see it with indirectly acting drugs, the ones that work by releasing a stored chemical.
Like ephedrine.
Ephedrine is the textbook case. It works by forcing the release of stored norepinephrine. If you give doses too close together, you deplete the stores faster than the body can make more, and the effect just disappears.
Let's shift gears to the mind. How much can a patient's belief or expectation actually modify a drug's action? We have to talk about the placebo.
The placebo is fascinating. It's an inert substance, a sugar pill, that works purely by psychodynamic means. There's no active chemical, but the belief in it is biologically active.
And that belief can cause real, measurable physiological changes.
Absolutely. The act of getting a placebo, especially from a confident doctor, can trigger the release of endogenous opioids and endorphins in the brain. This creates genuine placebo analgesia, real pain relief. We know it's real because you can block it with an opioid antagonist like naloxone.
So they're used in trials as a control, but do they have a rational place in actual clinical practice?
Their role in day-to-day therapy is very limited because the effect is so variable. But they can be used to manage a patient who strongly believes they need a pill, but, in the doctor's expert opinion, doesn't need an active drug. The prescriber's confidence reinforces the effect.
And the flip side of this is the nocebo effect.
The nocebo is the negative counterpart. It's the harm caused by negative expectation. If a patient is anxious, pessimistic, or has lost faith in the treatment, that negative expectation can actively undermine the therapeutic effect of a perfectly good active medication. The mind is powerful.
All of these variables really have to be unified under the idea of rational use of medicines. The World Health Organization has a definition for this.
They do. The WHO says rational use means patients get medication that's appropriate for their clinical needs. in doses that meet their individual requirements for an adequate period and at the lowest possible cost to them and their community.
Sounds simple, but getting there requires ticking a lot of boxes.
6 key criteria. It has to start with an appropriate indication. Then, selection of the appropriate drug based on efficacy, safety, suitability, and cost. Then the correct dose, route, and duration. Then ensuring it's for the appropriate patient with minimal contraindications. Then correct dispensing with clear instructions. And finally, adequate monitoring.
Yet, irrational prescribing seems to be a huge problem. What are the common mistakes?
Oh, we see some really problematic patterns. Using a drug when none is needed, like antibiotics for a viral cold, compulsive co-prescription of unnecessary vitamins or tonics, selecting the wrong drug, like giving a beta blocker to an asthmatic.
And the irrational use of combinations.
Yes, the unnecessary use of fixed-dose combinations. And critically, unsafe drug use, like using corticosteroids for a simple fever or prescribing only one antitubercular drug, which just guarantees resistance.
And the impact of that is profound. Increased mortality, microbial resistance, financial waste, loss of public trust.
To fight this, prescribers have to follow a logical, stepwise process. First, establish the diagnosis, then define the therapeutic goals, then select the drug class and the specific drug based on 4 pillars: efficacy, safety, suitability, and cost.
And the final crucial step in that chain has to be patient communication.
Communication is therapeutic. The prescriber has to explain the expected effects and, crucially, when to expect them. They need to discuss common side effects honestly without scaring the patient, and they must provide specific, actionable instructions like not drinking alcohol with metronidazole or not driving while taking a sedating antihistamine.
We've now established why choosing the right drug at the right dose is so complex, but none of this can even happen unless a molecule survives the massive multi-year process of drug development.
It is a truly monumental undertaking. From the initial discovery of a compound to its final marketing approval, the process typically takes at least 10 years and costs a staggering $500 to $1,000 million US.
$1 billion. That's why only huge pharmaceutical companies can really do it.
Exactly. The commercial risk is just immense.
So the pipeline begins with preclinical studies. This is all before it ever gets near a human.
Right. Once a potential compound is identified, it gets put through intensive testing in animals, usually one rodent and one non-rodent species, like a mouse and a dog, to characterize its full pharmacological profile.
What are the key stages of this preclinical testing?
It's multi-layered. You have simple screening tests to confirm activity. You have tests on isolated organs or cultures. You use animal models of human disease, like rats that are bred to have high blood pressure. And you do full systemic pharmacology studies to see its effects on all the major body systems.
And this is where you get the first look at its pharmacokinetics.
Yes, how the animal body absorbs, metabolizes, and excretes it. But the absolute focus at this stage is safety. So extensive toxicity testing.
How is that broken down?
It's rigorous and time-based. Acute toxicity determines the LD50, the dose that's lethal to half the animals. Subacute toxicity is repeated dosing for 2 to 12 weeks. And chronic toxicity studies go for 6 to 12 months and often run at the same time as the first human trials.
And then there's specialized tests for long-term safety.
Yes. You have to test for effects on reproduction and fetal development, that's teratogenicity. You test for genetic damage, that's mutagenicity. And you test for its ability to cause cancer, carcinogenicity, which can require whole-life exposure in animals. Only a compound passes all of this can the IND license.
And that transition from animals to humans is where things get ethically very complex.
Every single clinical trial has to be approved by an independent ethics committee. The whole framework is built on 4 pillars: autonomy, the participant's right to choose; beneficence, the motive to do good; non-maleficence, the obligation not to cause harm; and justice, fairness in who gets selected.
So let's get into the structure of the human trial, starting with a non-mandatory emerging stage, Phase 0.
Phase 0 is a clever strategy to reduce cost and time. It's a microdosing study designed to catch any bad human pharmacokinetics really early on.
How small are these microdoses?
Tiny, often 1/100th of the predicted therapeutic dose. They're subpharmacological, so they're not expected to have any effect, good or bad. You need incredibly sophisticated instruments to even track them in the body.
Then the official journey begins with Phase 1, human pharmacology and safety.
This is the first time the drug is given to humans. It's a small group, 20 to 80 people, usually healthy volunteers. The focus is almost entirely on safety and tolerability and establishing the dose range. You start at an incredibly low dose and slowly work your way up.
And if it's safe, you move to phase 2, therapeutic exploration and dose ranging.
Right. Now you're working with 100 to 500 patients who actually have the disease. The primary goal shifts from safety to proving therapeutic efficacy. Does it work? This phase determines the effective dose range and finds the ceiling effect, where more drug doesn't give you more benefit. This is where most drugs fail.
If it succeeds, it goes to phase 3, therapeutic confirmation and comparison. This is the big one.
This is the definitive proof stage. You're talking 500 to 3,000 patients across multiple centers. It's almost always a comparative study against the existing standard of care. The goal is to establish the drug's definitive value. The data from this phase is what goes into the new drug application.
And even after it's on the market, the development isn't over. There's phase 4.
Phase 4 is post-marketing surveillance. Now that the drug is out in the general public, the patient base is enormous. This allows you to detect rare, uncommon adverse effects that were just missed in the smaller trials.
And this is also where companies have to study the groups they excluded from the earlier trials.
Correct. Specific trials are now mandated for special populations like children, the elderly, or patients with organ failure to make sure the dosing modifications we talked about earlier are properly validated.
So to make sure all this data is sound, the trial design has to be the gold standard, the controlled trial.
Absolutely. The crucial piece is the control group, which gets either a placebo or the existing standard treatment, the active control.
And there are different designs for this?
Yes, you have the parallel group design, where you have separate test and control groups, or you have the very efficient crossover design, where the same subjects get both the test drug and the control drug at different times. They serve as their own controls.
That sounds great for minimizing individual variation.
It is, but you can only use it for stable, chronic diseases. The core challenge in any trial is eliminating bias from both the participants and the investigators.
And there are 2 essential strategies for that.
Randomization and blinding. Randomization means you use a random process to assign subjects to groups. This prevents anyone from consciously or unconsciously skewing the results.
And blinding means you conceal the nature of the treatment.
Right. In a single-blind trial, the subject doesn't know what they're getting. In a double-blind trial, which is the highest standard, both the subject and the investigator are unaware. This means the test drug and the control have to be physically identical in every way. The code is only broken at the very end.
So the absolute gold standard is the randomized controlled double-blind trial, the RCT.
The RCT provides the most credible, irrefutable evidence. Without it, the evidence is just considered too weak for wide clinical adoption.
So once all this evidence is collected and published, the medical community uses a hierarchy of evidence to figure out how much weight to give it.
Yes. Grade 1 is the most reliable. Systematic reviews and meta-analysis. This is the strongest basis for making clinical decisions. Grade 2 is a single, well-powered RCT. Grade 3 is less reliable things like open-label trials or observational studies. And Grade 4 is the least reliable, just case reports and anecdotes.
Let's look closer at that grade I standard, meta-analysis. What does pooling the data actually do?
A meta-analysis pools the raw data from several similar RCTs. By making the total number of subjects much larger, it dramatically increases the statistical power of the conclusions. It can help resolve uncertainty when individual trials might have conflicting results.
But there are criticisms, right, like publication bias.
Oh yes, the big one is publication bias, where trials with negative results are less likely to get published, which can skew the pooled results. But despite those issues, meta-analysis has fundamentally changed medical practice, like providing the overwhelming support for using beta blockers in heart failure.
And what about grade 3 evidence, like cohort studies?
Cohort studies are observational. The investigator just observes what happens. They compare events in users versus non-users of a drug. They're often used after a drug is on the market to find uncommon adverse effects that were too rare to show up in the formal trials.
They require long follow-up periods.
They do. Contrast that with a case control study.
Which are used mainly to look for an association with a suspected rare adverse event.
Exactly. Researchers work backward. They find people who had the rare event, match them with a control group who didn't, and then look back at their histories to see if drug exposure was higher in the affected group. They're fast and cheap, but they don't prove causality, they only strengthen an association.
So today, we have really navigated this immense journey of pharmacology, from the subtle influence of a single gene to the monumental 10-year effort for drug approval.
We've seen that the standard dose is just a starting point, a population average that has to be ruthlessly individualized based on a patient's age, their unique physiology, and the health of their liver and kidneys.
And we now have a much fuller appreciation for the rigor that underpins the medicine you take. The necessary trade-offs, the ethical scrutiny of the clinical trials, and the hierarchy of evidence that ensures decisions are based on the highest standard of proof.
Pharmacotherapy is fundamentally dynamic. Optimal drug use requires this continuous synthesis of drug knowledge, patient status, and the whole context. The dose you take really is the endpoint of a high-stakes, highly individualized decision process.
So let's end with a final provocative thought that ties back to that genetic variability we talked about.
We noted that a huge portion of individual drug response is traceable to our genetics, and genotyping a person only has to be done once. So given that, how quickly will pharmacogenomics move from being a promising specialized tool to becoming the mandatory standard for guiding the initial choice of drug and dose for every single patient? And in doing so, finally rendering the whole concept of a standard dose a relic of the past. Think about the logistical, financial, and ethical changes that transition is going to entail.